I love you.
I'm not me.
You're all the lab, same lab, bro.
Coming up on next Thursday, the 6th. That is going to cover from the book
Oh, shit, fine.
chapters 14, 15, and 16.
This is all I'm talking for the testing, is
I
it?
don't know what's supposed to do. Oh, shit.
And then
Yeah, one more for this?
corresponding lectures,
Is it
like
pencil?
13,
Oh, yeah. Where's my pencil?
ABC, 14, whatever it is.
Oh.
So 14, 15, 16.
Chapters in the book. And then you find the corresponding lectures. Okay? So for a lecture exam next Thursday, the book chapters 14, 15, 16. For the lab practical, which will be 2 weeks after that, right? Or a week and a half after?
A week.
A week after, the 13th, that will be from labs 13, nervous tissue. So definitely 15, so labs 13, 14, and 15. And we're gonna see what senses we can fit on that, to that, lab practical exam. So possibly some of 16. Okay? And I would say that
16 is the spinal cord and spinal
lab.
Yeah.
Lab.
Oh, is it from the last one? Okay. No, it's only
Okay, so we're gonna head into the census tonight.
What did you get, Eddie? What'd you
What's
get?
up?
The light back
What do you want to
there.
get? Oh, that? Pumpkin spice. Pumpkin spice. I'm that basic. Yeah, you know. Can't tell me it's not good though.
It's gonna take this bag up. Looking at you. You take
Are
the
they
bag
rich?
up for me?
Oh, tape it?
I got
That she said I ripped it.
Did you rip it?
No, I didn't rip it.
Did you tear it up, bro?
I could tape it.
Okay, that's let's talk about the station.
Where's the record at?
Remember that we are picking up information about our interaction with our environment via our sensory receptors all over the place. And those are in the peripheral nervous system, our sensory receptors in the peripheral nervous system. And all of that via electrical impulses traveling across along neurons, their axons, right? And then changing into chemical messaging via neurotransmitter being released, synaptic cleft, which causes an electrical impulse in the next or postsynaptic, the one that comes after the synaptic cleft, that postsynaptic neuron. Or maybe there's a muscle there, it's gotta be the neuromuscular junction, right? Okay, so all that information about sensation is coming into the central nervous system. Again, our sensory receptors are allowing us to get feedback from the environmental outside of us, the environment outside of us, external, but also internal. What's happening internally in our internal environment? What sensations are happening? Vagus tripping, and you were, like we mentioned today, it could be faint. All kinds of things can happen. The vagus has something going on with it, that cranial nerve that travels to every organ. So we have our senses categorized as our special senses and our general senses. Our special senses are basically in our head. Our smell, taste, balance and hearing are both in the ear, and vision. And then our general senses. There we're talking about our touch receptors, receptors for pressure, vibration, temperature, proprioception, being able to know where my hand is in space, right? Without even looking at it, I know where it is. Okay, so our sensory neurons connect our peripheral nervous system to our central nervous system. Sensory neurons. Where at the ends of them will be receptors, picking up whatever it is they're picking up. And that gets delivered as electrical impulse along that neuron. And then into the central nervous system, some places are at the spinal cord, right? Sensory is going to be delivered to the neurons in the dorsal root ganglia. And then those neurons whose axons will enter into the dorsal root of the spinal cord, and either right there, the spinal cord will figure out what to do as a reflex. We don't have time to send it up to the brain to think about it. We need to do something quick. Or it'll go up to the brain, and the brain will figure out what to do and send out some motor commands along the motor neurons. Okay, so our special senses have special sensory receptors that are called complex receptors, okay? Because they are housed in special sense organs, okay? So our noses and our olfactory epithelium, right? We saw the olfactory nerve, where its receptors are along the superior nasal concha, right? The ethmoid bone in the nasal cavity, and the tongue, where we find the special sense organs, what? For taste. The taste bud. And then our eyes, where we'll see the retina. And our ears, we'll see what houses both the part of that special sense organ in there, the cochlea for hearing, and the vestibule for stabilization and equilibrium. Okay? That at the vestibule, that's what, when we spin around, we feel dizzy, there's something going on in there. Okay, so let's start with the eyes and vision. Our special sense, one of our special senses, okay, the most complex sense, you'll see the retina, the neurons are located for perceiving light and then translating that by sending it up to the brain to vision, to seeing objects. So this is how we process our surrounding information that's contained in the visible light around us. And we're going to change that light. We get a whole transduction, a change of light into acting potentials or nerve or electrical impulses traveling along axons. And all that's going to lead to the formation of some images. So a lot of our cerebral cortex is going to be involved in processing visual information. That special sense organ for vision is the eye. Okay? The majority of our sensory receptors are in the eye, collecting light and converting that into nerve impulses. Being able to distinguish shapes happens at the eye. Being able to distinguish colors is happening at the eye. So we have our eyeball, irregularly shaped hollow sphere, okay, where we can see the majority of its, not the majority, some of its anterior side, and not all of it. Most of it is buried behind or the inside the bony orbit. We have a lot of fat within the bony orbit that's giving some cushion to the eyeball. We know about those muscles that run out and enter and make the eyeball move. Okay? So we have this whole evolution of eyes, where we all have all of our animals on the planet have a common origin when it comes to eyes. Okay? And we have this shared genes that are devoted to vision, the PAX6 gene. Okay? Opsins are proteins whose genes are encoded in DNA across species with eyes. Opsins are going to be the proteins we find in our photoreceptors that absorb light. So the eye has evolved, right? From over 600 billion years ago, where animals just had eye spots. Okay? And then we changed to more, animals evolved to have kind of a pinhole, structure as an eye, like what you would see in a camera. Now we've evolved to have the eyes like we do, or like eagle eyes. And this evolution across species, and when it comes to eyes, is change, is adaptation to the environment around them. All of this has happened in parallel across species. Eyes have been evolving in different species along their own evolutionary tracks, but in parallel with each other. And our adaptations, the changes in the eyes are going to be due to what's required. When I need to see tiny rodents from way up in the sky, then I'm gonna have an eagle eye. Okay, the eye itself has its accessory structures, the eyebrows, the eyelids, the eyelashes, there are glands here. The conjunctiva, anyone had conjunctivitis? Pink eye? That's where pink eye is, would be the lacrimal apparatus, making our tears, our extrinsic eye muscles that we've already studied. The eyebrows, also called anatomically the supercilium, okay, are going to be on top of our supraorbital arches of our skull. Remember those? Have to shade from the sun, keeps things from rolling down into our eyes. The palpebrae are the eyelids, okay? That's the slit between the upper and lower eyelid is the palpebral fissure, okay? That area between the eyelids. And we have a muscle that's devoted to lifting up that top eyelid, the levator palpebrae superioris. And then we have the tarsal plates. These are connective tissues that muscles will insert that open and close the eyelid. So orbicularis oris will insert onto the tarsal plates, cartilage, connective tissue. Palpebrae superioris will insert onto tarsal plates, connective tissue. The eyelashes are called cilia, okay, lining our palpebral margins. There's a reflex there, the blink reflex. When something touches our eyelashes, the reflex is to blink. So here we are. If we take away the skin, leave the connective tissue and muscle, we'll see the tarsal plates of the eyelids onto which muscles insert that will open and close the eyelids. On our model, and we do have this model downstairs, yes. We've seen this model, the ones with the muscles, to borrow some from the other room. We have the upper palpebra and the lower palpebra, the upper and lower eyelids. And again, this connective tissue is the tarsal plates. Now, one can suffer from blepharitis, chronic inflammation of the eyelid. Okay, blepharitis. We have burning and itching, light sensitivity, all caused by a bacteria. Okay, and we have what's the skin glands. So the glands that secrete oils can be inflamed. Okay, and so you go to the doctor, get your medication. Keep your eyes clean, take your antibiotics. Trichiasis is the eyelashes that are running the wrong direction, usually inward towards the eyeball. Okay, so that's going to be misdirected eyelashes rubbing against the eye. Of course, that's going to be irritating. Electrolysis, removal of the eyelashes, might be the treatment for that. Okay, so we have the corners and the corners of the eye, the medial and lateral canthus. Okay, so the medial canthus and the lateral canthus. You see that as the pink on both sides, so you have the pink in between your medial sides of your eyelids where they kind of come together in the corners of the eye. Then we have the lacrimal caruncle. This is where we have a gland that produces a thick secretion that when it dries, we call that secretion sleep. Alright, so someone said you could sleep out your eye. That's that nodule that's in the medial canthus. So the medial lateral canthi are the corners of the eye. In our medial canthus, you have that pink kind of bulb. That is the lacrimal caruncle. And then we have the puncta, the superior and inferior puncta of the medial corners of the eyes. Those are the small holes. So you might have seen the hole that's kind of in the margin of your eyelid, in the corner, the medial corner of your eye. That is a puncta. We have one at the top and lower eyelid, and that's where when tears will flow into. And when we have an overflow of tears, they come out, right? But we're always having tears going across our eyes, and they will run into the puncta. Here we are at our medial and lateral canthi, corners of the eye. The medial canthus, we have the lacrimal caruncle that secretes a secretion that when it dries, we call it sleep. We have the puncta, superior and inferior puncta, which are the little holes that will be right there at the ridge of the eyelid and margin of the eyelid that receives tears. So on the model, you can see the puncta. The caruncle and the corners of our eyes, the medial canthus and the lateral canthus. Our glands are there to secrete secretions that are protective, okay, and also provide lubrication. The tarsal glands help to keep the eyelids from sticking together. So tarsal glands are around the margins of the eyelids. They're secretions, kind of oily secretion, lipid rich, that's keeping the eyelids from sticking together. Tarsal glands. On the model, you can see what's depicted as the tarsal glands. There are glands and then the ducts. So that's what I'm referring to. So the glands, those are the glands. This is for the tear duct. First, we also have some other glands, some ciliary glands. These are kind of located like under the skin of the eye, the ciliary glands. And we have our other gland, the lacrimal glands, are what produce our tears. Okay, and these are superior and lateral. So the superior and lateral portion of the eye is the lacrimal gland. It produces our tears. And then the conjunctiva is the epithelial lining of the eyelids. So we have the palpebral conjunctiva that lines the eyelids. Then we have the bulbar or ocular conjunctiva. Okay, so the palpebral conjunctiva that lines the backside of our eyelids will then fold back upon itself and line the white surfaces of the eye, the whites of the eye. And that's what gets infected in pink eye. So here that is the palpebral conjunctiva, that's lining the backside of the eyelid, folds back upon itself, and then lines the whites of the eye. That's the bulbar or ocular conjunctiva. The whites of the eye is called the sclera. Sclera is the whites of the eye. That's covered by bulbar conjunctiva. And then we see here the iris. That is whatever color it is, depending on your level of melanin expression. The more melanin, the more brown it is. Very little melanin, of any melanin expression at all in your iris, blue eyes, and then the pupil, into which the eye or the light travels. Okay? So when it comes to the conjunctiva, that bulbar conjunctiva, we don't have any of it covering the iris, or definitely not the pupil. Okay? So just the whites of the eye. Then our lacrimal apparatus, these are the glands and ducts that will secrete and drain or the tears. So the gland secretes the tears, and those will be then drained into the nasal cavity. Okay. So the lacrimal gland, again, is at the superior and lateral side of the eye. The lacrimal secretions are tears, a salty saline solution that also contains some mucus, antibodies to, if there's any pathogens in the eye, those antibodies will bind to those pathogens, labeling them for the immune system cells to come kill those pathogens infecting our eyes. Lysozyme is secreted in our tears, that's antibacterial. So we have this thin film that's 3 layers. You have the mucus layer, the watery aqueous layer, and then the oily lipid layer. It keeps everything, all that watery layer in place. That's what the tears do. Keeps our eyes moist and clean. Every time we blink, we are moving our tear solution, our lacrimal solution, medially. Every blink moves it medially so that it can drain. Okay? And again, we have excessive production of lacrimal secretion, we call that crying. And if there's a dysfunction, we call that watery eyes. Okay? So in our model, you've seen this. This is the lacrimal gland that produces the tears. It's on that other type of model in the room that we probably be pulling out now. Again, the tears are produced at the lacrimal glands. They flow medially over the eye, the surface of the eye, and then flow into those lacrimal punctus. Okay, those little holes. You can see that punctus if you look closely in the mirror. Everybody seen that in their eye? I did, right? You know what I'm talking about. Okay, and then there at the punctum, we enter into a little tiny canal, or canaliculi. See this word again, we've seen it before. Little tiny canals are called canaliculi all over the body. And then into a sac. And then we flow into what's called the nasal lacrimal duct. Oh, this sounds like describe the path of tears for me. Okay. Then we flow into the nasal lacrimal duct. And this duct will enter into the nasal cavity at the inferior nasal concha, that bone that's a bone all by itself. Okay? And so when our eyes get watery, all of this is going to happen. All that tear solution is going to head into the punctum, the medial corners of our eyes, and down into our noses. That's why when we cry, what happens in our noses? Runny nose. So you get a runny nose.
So when people have dry eyes, they're not producing.
Yes, we're either not producing, we're not producing enough tears. Yep. Okay, so here we are, the lacrimal apparatus, again, producing tears here, the lacrimal gland, that flow medially into the punctum, the superior and inferior punctum at the eyelids, down into the lacrimal canaliculi, here and here, and then into this lacrimal sac, here, and then down the nasal lacrimal duct, and into the nasal cavity at the inferior nasal concha. And on the model, this is represented there. The lacrimal canaliculi, taking us into the lacrimal sac, and down the nasal lacrimal duct, into the nasal cavity at the inferior nasal concha. So if there is an obstruction of that nasolacrimal duct, an NLDO, okay, which can happen with pink eye, okay, then you'll get a buildup, right, of the secretion that can't move down that the nasolacrimal apparatus. So you can get this NLDO with different dysfunctions, and massage it, push that, make it drain, or definitely probably get some type of medication to clear up the underlying cause, right, infection. So we remember our muscles, don't forget our muscles, superior rectus, lateral rectus, medial rectus, right? Superior oblique, inferior oblique, we won't forget those guys, those extrinsic muscles that move the eyeball. We're gonna pull out this small, as long as we can see everything here, I'll see, is on here. Hopefully, we'll get a depiction of the optic nerve that punctures out of the back of the eye. These are axons that are all the neurons in the retina. Their cell bodies are in the retina. And a layer of the eye that we're gonna go through. They puncture out the back of the eye as the axons do, as the optic nerve. Okay. And I'm gonna take a look at our model. Great, that's okay. This is a nice puzzle that shows us everything that's going on in the eye. A nice model. Okay, so one thing that we see in the body are layers that are called tunics. This body we can call layers the tunics. And the eyeball has its tunics as layers. It has a fibrous tunic, a vascular tunic, and a neural tunic. And the eyes are the only part of the brain that we can see. All right, so in our tunics, some of the things that we can see the cornea, that's in the tunic, the iris, the pupil, the sclera, the whites of the eye, the lens that bends light, the retina, that's where we're gonna have our neurons, those axons will make the optic nerve. The eye is divided into an anterior cavity and a posterior cavity, the eyeball. Okay, so if we look at a cross section through the eye and look at the tunics, we'll have the fibrous tunic that is made of the sclera, the whites of the eye. Then deeper to that is the vascular tunic, where we'll see blood vessels. There, we call this choroid, not to be confused with choroid plexus in the brain, but still having to do with blood vessels and so forth, like choroid plexus does. And then the neural tunic, that's where we're gonna have all of the neurons devoted to vision at our deepest layer. So the fibrous tunic of the eye is the outermost layer, lots of this connective tissue there. Some protection, but easy to damage. This is where our extraocular eye muscles will attach. The fibrous tunic consists of the sclera, the whites of the eye, and the cornea. Where the cornea is the clear part that covers where the lens is behind, okay? So we go from the sclera, the whites, to the clear cornea. Of course, it's going to be clear so that light can come in. But here's a nice cross sagittal section through the eye. And you can see the different layers, okay? Make up or within the entire sphere of the eyeball. Fibrous layer, the vascular layer, and then the neural layer is deepest. Here's how those axons of neurons will run out as the optic nerve. So on our model, the sclera and the cornea make up the fibrous tunic, sclera, and the cornea. The sclera is that white opaque region is this dense irregular elastic connective tissue. Okay, and it makes up the majority of the sphere, that is the eyeball, in the fibrous layer at least. Protects the eye, shapes it, again, where muscles attach. It will be continuous with the cornea and the dura mater of the brain. Posteriorly, sclera is continuous with the dura mater of the brain. And it's what will be punctured as the optic nerve heads out the back. So we see the sclera, some dense connective tissue, irregular connective tissue. Nice electron micrograph of that dense irregular connective tissue that makes up the sclera. The whites of the eye that's continuous with the transparent portion of the fibrous tunic, the cornea. So this is modified sclera. No blood vessels here, no opaqueness here, nice and clear, so that light can come through. And this is the bulbar conjunctiva will stop at the cornea, okay? This won't cover the cornea. So the cornea, behind the cornea, and therefore the cornea is covering the iris, which gives color to the eye, the pupil, right? The diaphragm within the eye that opens and closes to let light in. And it covers what's called the anterior chamber of the eye. The cornea helps to bend light as it comes in. So we want to bend that light towards the pupil. So the cornea helps to bend that light. Okay. There are pumps there, sodium pumps, embedded in the cell membranes of the cells making the cornea that help to keep the cornea clear. Lots of pain receptors there, even though reflexes occur here. It's able to repair and regenerate itself, and we can get a transplantation of cornea that will not be rejected. Why is that? Why do you think that that's the case? Many transplants can be rejected, right? Heart transplants, if you don't match the donor well enough, it can be rejected, right? It causes an immune system attack on that transplanted organ. But we can transplant cornea from person to person, and it would be a person who's recently passed away. His cornea will be quickly removed and transplanted to someone who needs. But why wouldn't it be rejected? Why do you think? Back to say again? Not that it's you said identical enough?
In everyone, she said.
Oh, in everyone? Not that it's identical enough in everyone. Let me see where it says
Is
it.
it because it doesn't share blood?
Well, it's because it's avascular. I'm trying to find where it says that. So there isn't any blood running in it. So therefore, we're not gonna have anything that, there we go, avascular. Okay, so there's nothing there that the immune system can see as foreign. Okay, and the tissues are similar enough, as long as you don't have anybody's blood running in there too, then the tissues are similar enough that the immune system from one of us to another of us will recognize those cells and the proteins that they make. Yes, because there's no blood, we don't have anybody else's proteins in the vicinity. You have a question?
I have a question about the sodium pumps in the cornea. So if they're not working, would that be responsible for like a cataract because it's not clearing it?
So cataracts is, we'll talk about it. Not certain it is the sodium pumps that are doing it. It's definitely
Would it cause a statin not working?
I'm not sure if it's the sodium pumps. But it's definitely a fogging up of that area. of the cornea, histologically. Then our vascular layer is the middle tunic of the eyeball. This is where we find the blood vessels, the lymphatic vessels, the intrinsic muscles of the eye. There's some muscles in here we have to learn of the eye, okay? Those muscles that help us bend and stretch the lens of the eye in order for us to focus far away and focus up front, okay? That's the bending of the lens. That's due to some intrinsic eye muscles, muscles inside of the eye. Okay? The vascular tunic is regulating the amount of eye coming in. So we're controlling the shape of the lens here. Okay? We'll talk about the aqueous humor here, but this is where the aqueous humor is secreted and reabsorbed. The aqueous humor is what keeps the eyeball structured. Okay? The vascular tunic has its three regions. The choroid region, the ciliary body is there. So the choroid, the ciliary body, which is muscle, and the iris. All a part of the choroid. Now we're here at the choroid layer. Or excuse me, at the vascular layer, vascular tunic, where we have choroid. Then we have the ciliary muscles that are anchored via the suspensory ligaments to the lens. And as they contract, they stretch the lens, they relax, the lens can bulge up, and that will change our vision. And the iris in front of the lens, where there's that pupil. So when we look into our model, we will see the vascular layer. This consists of the choroid, where we have all the blood vessels. That's deeper to the sclera. The iris is a part of the vascular layer. And then the ciliary muscles. The muscles are here, but we also have these processes and ligaments that help the suspensory ligaments attach the ciliary muscle to the lens. That's not here. The lens will be sitting here. Vascular tunic, and actually, at the choroid, that's where we have the blood vessel. This is supplying the blood to all of the layers of the eyeball. It is between the whites of the eye, the sclera, and then the deepest layer, the neural layer, particularly the retina. The choroid itself is pigmented. It has brown pigment, yellow melanin, all over the place, and that brown pigment is helping to absorb light. Okay? So that we don't have a whole bunch of light just scattering inside of the eyeball to disrupt the photoreceptors, okay? And that can cause blurry, if you have scattered light, that's gonna make things blurry, okay? So the choroid is darkly pigmented, brown pigment, in order to absorb any light that isn't any scattered light, okay? We only want that focused light to come in and hit the retina in the right spot, not all kinds of light from everywhere. Then we would have confused vision. So in our model, this picture up there. Oh yeah, this is guy we have. We have this one. We can see the choroid. Nice vascular layer, the choroid, and pigmented. Blood vessel. And then the ciliary body. This is tissue around the lens, where we have the ciliary muscle that has processes. And those processes have attached to them the sensory ligaments. And those sensory ligaments are suspensory ligaments. Those are then attached to the lens. The ciliary muscle, now we're talking about smooth muscle. And the ciliary processes have capillaries running through them, They secrete aqueous humor that we're gonna talk about. And those suspensory ligaments, again, anchor the ciliary processes of the ciliary muscles to the lens. Okay, so here's the lens. Here are the suspensory ligaments. Here are the ciliary processes of the entire ciliary muscle. The lens is back here, deep. Ciliary muscle, smooth muscle encircling the lens. It has processes that have attached to them the suspensory ligaments that are in turn attached to the lens. That again, the lens is back, is here. Suspensory ligaments that are attached to the ciliary muscle. Goes around the eye. So if we look at this histologically, take a cross section, put it on the slide, we can see lens. Here is iris in front of the lens. Here is cornea. And down here will be sclera. This opening here is the pupil. So this is the front of the eye. Light will come into the pupil. Here is the ciliary muscle and its processes. The suspensory ligaments would be anchored from the processes up to the lens. You can see the suspensory ligaments here that leave the processes and then attach to the lens. This makes the ciliary body. The iris, again, is whatever color, depending on your level of melanin expression in your iris, from blue, no melanin, to green, to hazel, to brown. Smooth muscle is going to contract and change the diameter of the pupil, the opening at the center of the iris. That's how the light is regulated, the amount of light is regulated coming into the eye. There's some muscle, the sphincter papillae muscle that closes the pupil. And then the dilator papillae muscle that opens the pupil. Again, we will have our eye color depending upon our level of melanin expression. That's all that it is. Melanin. Hello, melanin. Or for some reason. Okay, and then the eye dilation, the dilation of the pupil is controlled by the nervous system. We have the branches of the nervous system. The goal is to fight or flight, the sympathetic nervous system. That's gonna open that pupil up and let light in. So we get all the light we need in order to book if we need to run. Okay? And the parasympathetic kicks in. That will close down the pupil. You don't need all that. We're in a relaxed mode. We're feeding, we're possibly breathing, whatever we're doing, we don't need a whole bunch of light to do. So the parasympathetic is in charge, but it can get overridden by the sympathetic if need be. And that will dilate, the dilator pupillae muscle will open up the pupil, and the sphincter pupillae muscle will constrict. We can see how those muscles run. Okay, there in our model is the iris and the pupil at the center. So the cornea and the iris sit in front of the lens. So here's the lens and the cornea, there's the pupil. And here's the iris. Light comes on in. Sclera back here, ciliary muscle, the ciliary body is the ciliary muscle with its ciliary processes. With here we can see some suspensory ligaments running from the ciliary processes and attaching onto the lens. The lens is the only structure of the eye that is not a part of any of the three tunics, not a part of a layer of the eye. It's transparent, biconvex, so convex on both sides, okay, bulging out, flexible, avascular, don't want any blood vessels in the way of light coming in. This is where we have accommodation, meaning it's going to flatten or bulge in order to accommodate the light coming in and bend the light back onto the retina. So it's gonna change shape in order to focus light onto the retina. And it's that ciliary body that's changing the lens shape to focus light, to help us focus on things that are near or far. And sometimes we need help with glasses. The lens has its epithelium, its anterior surface has epithelium, is an epithelium called the lens epithelium, and then there are lens fibers that make up the deep bulk of the lens, made of a protein called crystalline. One good example I gave in my genetics class is that all of our DNA is encoding every gene that is expressed, that we express, but it depends on the cell type as far as what proteins actually get made, what genes are actually expressed, and therefore the proteins that are made. So our liver cells, the DNA in those cells have the gene for crystalline, but because they are liver cells, they don't express that gene, and therefore they don't make the crystalline protein, and vice versa. Our lens cells they express the crystalline gene, and therefore make the crystalline protein, but they don't make a protein that is specific to the liver. So different cell types all have the same DNA sequence within their nuclei, but they each are going to express specific genes, therefore make specific proteins. And that's what makes cells types different from one another. So our lenses cells are producing crystalline to make the lens. So we have the lens behind the iris. Here's that ciliary body that is the muscle. Its processes that are connected to the suspensory ligaments that are in turn connected to the lens. On this already. Okay, so cataracts. It's actually not the cornea, it's the lens that is opaque. It's a cloudiness of the lens. So, and this is due not to sodium pumps or anything that aren't here, but there's a lack of nutrients coming to the lens itself. And different things can contribute to that, right? There we go. We can get that replaced by with an artificial lens. Okay? So cataracts is a clouding of the lens that will then affect vision. So it's not the cornea that's affected. Okie dokie. And okay, let's head to the neurotunic. All right, this is where we have vision happening at the retina, also called the sensory tunic. This is where you have our sensory receptors. This is the brain, okay, in the eyeball, light sensitive tissue. That light information is going to be sent into the central nervous system. The neurotunic consists of layers of neurons and synapses between these layers of neurons. Meaning one layer of neurons sprinkles out neurotransmitter that affect the next layer of neurons. They then sprinkle out neurotransmitter that affect the next, and that whole sprinkling of neurotransmitter equates to, electrical impulses traveling down the axons of these neurons. They basically pass that sensory information to each other that way. Alrighty, the neural tunic consists of the pigmented layer and the neural layer. So we have an outer pigmented layer and an inner neural layer. Okay, so now we're at the neural layer, also called the retina. The most inner layer of the eye, the neural layer. That can, the neural tunic that consists of the outer pigmented layer or pigmented retina, and then an inner neural layer. Okay? So in this model, this will be the most inner part. This will be underneath this, the pigmented retina is underneath the neural retina. So the neural retina is the most deep layer in the eye. So the pigmented layer is the outer layer of the retina. It's anchored to the choroid. Okay, so it is attached to the choroid. There's a choroid, so this would be where we find the pigmented layer of the retina. And then here would be the neural layer of the retina, deepest. It's pigmented too because it's helping to absorb any scattered light. We don't want any scattered light to mess up our vision, our acute, accurate light headed back to the retina. You don't want anything messing that up, especially any scattered light rays. Okay? This is where we get vitamin A stores. This is why Grandma said to eat your carrots for your eyes. The vitamin A is needed by photoreceptors that are in the neural layer. So that vitamin A gets stored there for them in the pigmented retina. Okay, so this is the inner layer here, the neural layer that's lining everything, the backsides of everything here, and this is the pigmented layer of the retina. Lines the everything. Okay. So there we see pigmented retina.
Is it something about that small thin line?
This dark line, yes. Pigmented retina. And then the neural layer of the retina, the deepest layer in the eye. This is where vision occurs. It's transparent. It extends anteriorly and to the posterior side of that ciliary body. It forms the serrated structure, the ora serrata. Okay, the serrated junction between the neural layer of the retina and the ciliary body. So let's see that. Here is the ora serrata. Serrata. Serrata, after these double R's. There it is, that ciliary body covered with pigmented retina. And then the neural retina starts after this ora serrata. Okay, let's get our bearings here. Here's that ora serrata. This is ciliary process of the ciliary muscle. Suspensory ligaments attached. The lens will be sitting here. All of this is lined with covered with pigmented retina. And that pigmented retina even goes back underneath this neural retina. And not underneath it, it's anterior to it, okay? The neural retina is deepest, okay? Deeper to the pigmented retina. Everybody following me on that? Okay. All right, then neuroretinal. Got different types of neurons here, ready to do their thing so that we can see, that we can make, alright, we can make some images from what the light that's coming in. There's light bouncing off of all of you that's going into my retina, and from that, I'm making images of you. Okay? At my photoreceptors, to my bipolar cells, finding my ganglion cells. Uh-oh, ganglion. The ganglia, in the peripheral nervous system, signals are spread from the photoreceptors to the bipolar cells. So they're sprinkling neurotransmitters that binds to the bipolar cells. And the bipolar cells get excited. And the electrical impulse travels down to their axons and sprinkles out neurotransmitter from them at the cell bodies of the ganglion cells. These guys are going to go on, their axons go on to become the optic nerve and ganglion cells. So we have that information, of sensation, of light coming in, bouncing off of objects in our environment, coming in, they hit the photoreceptors, that light. Then it's going to be transduced into an electrical impulse at the photoreceptors, changed into an electrical impulse, the light. And then transfer, ganglion cells, optic nerve. Okay? So we have millions of photoreceptors that consist of the rods and the cones. And that's where light energy gets changed or transduced into electrical impulses that will cause neurotransmitters to sprinkle out the ends of axons. Again, the ganglions, the bipolar cells are a layer in between the photoreceptors and the ganglion cells. The axons of the ganglion cells will all converge together in one place called the optic disc. We call that our blind spot. That's one place we have no vision, is where the optic nerve, the axons of all the ganglion cells, puncture out the sclera, the back of our eye. Okay? The whites of our eye, back there. And that's the optic nerve, headed towards where what crosses at the optic chiasm? Don't tell me optic nerve. The medial fibers of the optic nerve will cross. Those lateral fibers keep going back on their same side. Okay? And then that optic nerve is going to conduct all those action potentials or those electrical impulses along the optic tracts to the thalamus. Thalamus will relay that along projection fibers back to the optic occipital lobe. All right, so here we are. Let's understand what we're looking at here. Our layers of neurons. Okay. Here's the pigmented layer of the retina. Okay. And here's the neural layer. The neural layer consists of the photoreceptors first, where we have rods and cones, rods and cones, rods and cones. And then we have the bipolar cells that are received or get sprinkled with neurotransmitter by the photoreceptors. Then we have the impulse traveling down them. And then they sprinkle out neurotransmitter that goes and affects the ganglion cells. And all of the axons of the ganglion cells all converge together to make the optic disc, or the optic nerve. So, what happens, light actually comes in, so you can see the pathway of light. Light actually comes in this way, but it's not going to do anything until it hits the photoreceptors, okay? Because this is the deepest layer, this is the deepest layer, the neural layer, okay? Of the eye, the retina, okay? The neural layer of the retina. So the ganglion cells, their axons, are the extreme most deep portion of the neural layer, okay? The very, if we could, and I would like to dissect some eyeballs. We can cut open the eyeball and then we touch inside that eye, we will be touching where the axons of the ganglion cells are. That's the deepest part of the neural retina. But light doesn't disturb these guys. Light's gonna come on in. It won't mess with these guys. It won't mess with the bipolar cells. It's the photoreceptors that will get triggered by light. They will get triggered, and then they will pass information back this way. All right, there's the choroid. That's more superficial to the neural layer, excuse me, the pigmented layer, no, the choroid is here. That is more superficial, this more superficial tunic to the neural layer. The neural layer consists of the pigmented retina, this thin region here. That is more superficial to the neural layer, the neural retina. This is the deepest we can get into the layers of the eyeball. Light comes in, bypasses out all those fellas until it gets to the photoreceptors, consisting of rods and cones. Okay. Some of here's the nuclei of the photoreceptors. The rods and cones make up the photoreceptors. So here's the cell bodies of those neurons. They have an extension, or extensions, that embed themselves in the pigmented layer of the retina, where their vitamin A is stored. And they get to soak up vitamin A that way, that they need, the photoreceptors. And then they have another extension that connects them, or from which they sprinkle out neurotransmitter and affect the next folks, the bipolar cells. Here are their nuclei, their cell bodies. And then they sprinkle out neurotransmitter onto the ganglion cells. Here are their cell bodies, the nuclei of ganglion cells. And then their axons go on to make the optic nerve. That's more of the same. The ganglion cells, the bipolar cells, and then the photoreceptors, and then the pigmented retina. Histologically, light comes in, bypasses is here. The light stimulates these fellas. They have an extension tucked into the pigmented retina. We have vitamin A that they get to soak in so that they can keep working. But light excites them, and they take light, they change it into an electrical impulse that travels and affects the bipolar cells. The electrical impulse travels, changes into neurotransmitter stuff, and then that affects the ganglion cells. This is where we're gonna finally get that sensory information sent back into the brain. Same thing here, just beautiful fluorescence. Nice fluorescence. Look at the choroid layer where you see blood vessels. Very nice. The pigmented layer of the retina, the neural layer, you see the cell bodies, the nuclei of the 3 layers of neurons here. The rods and cones are modified sensory neurons. These are the receptor regions. Okay, so they are picking up, they're getting affected by light. There's photopigments here. The rods have one particular photopigment. These perceive gray tones. No color vision at the rods. Picking up gray tones here. But at the cones, here we have high resolution detailed color vision happening at these neurons. And the rods, gray tone, fuzzy, no color, no sharpness, but still necessary. Those cones though, man, that's what you want. You want that sharp color vision. The photoreceptors can be damaged. Okay? And if the retina detaches, the photoreceptors will start to degenerate. Okay, so we don't want that. If we get intense light, that can then destroy our photoreceptors, so we don't stare at the sun. Okay? Damage our photoreceptors. So here we are. Let's get our bearings. Here is the pigmented retina. Here are rods and cones. They have an extension that's embedded in that pigmented retina so they can get vitamin A that's stored in the pigmented retina. Here are the cell bodies of these neurons, and here is their this inner fiber They have an outer fiber and an inner fiber. This inner fiber is what then will sprinkle neurotransmitter at the next fellas, the bipolar cells. Not touching, but there's gonna be a synaptic cleft between them. The rods and the cones. The rods and the cones. With their extensions down embedded in this pigmented retina. The rods and the cones. Nice electron micrograph of rods and cones. Color blindness is a problem with the cones. So you have red, green color blindness where you can't tell red or green. You lack the red and green cones, those cones that decipher red and the color red and the color green. And so you can't make those colors out. They look gray. This is inherited, an X-linked inherited condition. You usually find it in males, okay, because if it's on the X chromosome and on mom's X chromosome, there's a mutation in the genes that encode the red and green cones. Then a male that will receive just one X from mom won't have another X to compensate from dad. They'll have a Y from dad. Okay? And so they end up with color blindness. Females, on the other hand, might receive from mom an X that has a mutation in the genes that encode the red and green cones. But dad's X might have the gene that's just fine, and that will compensate for any mutation on mom's X. As long as you got one X with a good gene, okay, so it's more likely that a female won't have color blindness. Now, if she happens to get from mom and dad, then a female can have color blindness. Okay. Night blindness is when the rods are impaired. Okay, because the rods are all about gray, dim, fuzzy light, okay, vision. And so if the rods are impaired, then you'll have trouble seeing in very dim light. Okay, and that could be because you're not eating your carrots too. Okay, I'm gonna leave it there. We're gonna come back to more regions of the eye next
Clinical
time.
Pro. Just those examples of disease.
Okay.
Yeah, like some kind of impairment.
Huh.
That's what she like to
So there was a sign-in sheet for Lab B, all lab B's over here are signed in. And the sign-in sheet